Welcome to this class on plant protoplast culture, the inoculation chamber where you do all your activities. What is protoplast? Protoplast is a naked cell, a cell without a cell wall. The term was first used by Hanstein in 1880. This is the isolated protoplast. There is a difference between protoplast and spiroplast. There is another word. Protoplast lacks the cell wall. It is totipotent. That is very important. It is capable of developing into a new plant completely and allows the entry of foreign DNA and the organelles. That is what is most important, but highly sensitive to osmosis or osmotic solution. So we have the significance of protoplast. Why do you do protoplast? Two important methods, two important reasons. One, to bring about somatic hybridization. The other one is to introduce the foreign DNA and foreign organelles. So somatic hybrids are produced. Hybrids also can be produced. We will learn it later. What are hybrids? What are hybrids? We have an intact plant cell with cell wall. We use the enzymes, and osmoticum is used. Select the young plant. Take the healthy leaf, peel the epidermis, take the peeled segments, keep it in plasmolytic solution, allow the cell walls to digest in the enzymes, centrifuge it. Repeatedly you can do it. You get the pellets of protoplast. Here you can introduce the genes and organelles. You get the regeneration of cell wall, cell division begins, you get clumps of cells. You can use these colonies for producing secondary metabolites, or you can use them for multiplying the plants. This is the purification of protoplasts. The protoplasts that are isolated by either enzymatic method or mechanical method have to be purified, and you can see in the last test tube pellets of the protoplast. So this is the biotransformation via protoplasts. The protoplasts can readily take up foreign DNA and foreign organelles. Isolation of protoplasts is either done by mechanical method or enzymatic method. Enzymatic method, we have two types. This is the mechanical method where you cut the cell with the knife and keep them in plasmolytic solutions, and the protoplasts are isolated, keep them in the plasmolytic solution. The protoplasts are released. Mechanical method was first used by Clarker in 1982. Cut the cell with the sharp knife into small pieces, plasmolyze them, release the protoplast, again keep it in the deplasmolytic solution. So what is most important is the osmoticum, alright, and also you have to keep them in plasmolytic solution, which is 1 molar sucrose. Micromanipulator is a modified microscope with the help of which you can actually see and break the cell wall and release the protoplast. So protoplast, there is some problem sometimes encountered along with the isolation of the protoplast mechanically. Sometimes the plasma membrane breaks because of the high concentration of the solutions that are used. There are drawbacks. Only vacuolated cells can be used for this. Low yield is there. Coming to enzymatic method, Cocking 1960 used it first. Osmotic shrinkage is minimum, the yield is high, it is not as tedious as in the case of mechanical method. There are again two steps here, direct method and a sequential method. Direct method, one combination of enzymes are used, all enzymes are used together. In sequential method, enzymes are used in two steps, direct method and sequential method. In both the cases, take the leaf, sterilize it, remove the epidermis. And then the epidermis is cut into pieces. Allow it to undergo plasmolysis. Plasmolysis has to take place by using the high concentration of sucrose, 1 molar sucrose. So in direct method, you are using pectinases, cellulases together. Protoplasts are readily released. You get the isolated protoplast. In the indirect method, what you are doing is, in the first step you are using pectinase, centrifuge, release the isolated cells, and then these isolated cells in the second step again are treated with the other enzyme called cellulase. First, middle lamella is broken, in the first step. Second step, cell wall is broken. So finally you get the isolated protoplast. Direct enzymatic method, you get protoplasts in the form of pellets. They have to be washed in sorbitol and before centrifugation. This is called protoplast washing solution, and this is what is also called osmoticum. So these are steps used in protoplast isolation. There are 8 steps. Sequential method, you have 2 combinations of enzymes. One is mixture A, mixture B. These 2 are used in a sequential manner. First you use mixture A, second you use mixture B. And they have to be washed in protoplast washing solution, which is nothing but 13% mannitol, also called osmoticum, and a specific pH is used. These are the isolated protoplasts. So in the left hand side you have a vial with a red cap. Large protoplasts, we don't use them. Optimal protoplast, that is what we are using. At the bottom you get the debris, dead protoplast, cell wall and all those things. So this is how you get the protoplast. When you finally centrifuge it, the last stage of centrifugation, you get the isolated protoplast. You all can see that protoplast. Sometimes you also can use some dyes so that they can appear in bright color. Finally, you have to purify the protoplast. 3 kinds of things have to be removed. One is undigested material, that is debris, mostly made up of dead protoplast and cell wall, burst protoplast and enzymes. So all these are used by using filtration. Thank you very much for watching my video. I thank all those resources from where I collected this information. Like, share and subscribe so that you can get the alerts, so that you can see whenever I release the next video or next e-content, you can see it